Okay, so questions 9A and B, we're going to try to do together. I know there's not a lot of room for B here, but we're going to try our best. So this type of question, toughest questions you're going to see in my opinion, because you're looking at two genes that are interacting on separate chromosomes, and one of them is sex-linked, one of them is just autosomal. So you got a lot of stuff going on here. Now, because it's so difficult, we've actually put the legend in here. So it says in fruit flies, eye color is determined by two genes located on different chromosomes. Presence of a dominant B on one autosomal chromosome and a dominant X with a capital V on the sex chromosome results in wild type red. And they give you the legend there. So let's just transfer that just to make it stand out from the rest of the question here. X with a capital V, that's going to be red. Okay, wild type red. Now they're saying a recessive little b in a homozygous condition along with a dominant X with a capital V gives us brown eyes. And they've given us the legend here, so let's just transfer. That's all we're doing. Very difficult question, but that's why we've kind of given some of the genotype or some of the legend here embedded in the question. That's going to be brown. And then it says a present of a dominant B along with a recessive sex-linked gene X with a little v. Now those are supposed to be little v's. I couldn't show a little hook on them. So it's going to produce you, little v in a homozygous condition is going to produce you vermilion eyes. And there's the legend there. Have to have a capital V, doesn't matter what the other allele is. But if I have two little v's, now I'm going to show the little hooks on them so I don't get them mixed up with my big V's. That's going to be verm. I'll abbreviate that, but vermilion. And if I have the presence of brown, which is two little b's, and vermilion in a homozygous condition, so they're saying X with a little v, X with a little v, that's going to give me white eyes. Okay, so lots of that question, that's why you were given the genotype here. If you see a question like this, which I don't think you are on one of the exams, they will have to help you out a little bit by giving you some of the legends embedded in the question. So let's go through questions A first of all. It says a wild type female with this genotype is crossed with a white-eyed male. Okay, so white-eyed male, I guess we could, these are all, so white-eyed male, let's add on to this legend. If I have little b, X with a little v, and a Y, that's going to give me a white-eyed male. Okay? All right, so now let's go through this question here. We're going to just do a Punnett square. I'm going to put it up here so I have lots of room for B, because actually B is a longer question. And we're just going to do the same folding system that we did with our regular dihybrids. So we have a capital B with a capital X with a capital V. Okay, and everything else is a repeat. So that's it for the female. Female goes on top, male goes down the side. Little b with the X with the little v. Put the little hooks. And then we also have the little b with the Y. But everything else is a repeat. So we've now taken this Punnett square and we made it into two blocks. Bring them together, capital B, little b, X with a capital V, show the dominant first, and a little v. So as long as I have a B with a big V, I don't care what the other alleles are, this is going to be wild type red. Do the other one, capital B with a little b, X. Now this is actually a red female, so let's put the red female in there. And again, when there's sex link, we want to isolate the males and the females. This one is a Y, and same thing. As long as I have, forgot my big V there, as long as I have a big B and a big V, it's going to be red. But if there's a Y present, we know it's going to be a red male. And that's our question. 1, 1 ratio, 1 being red female, 1 being red male. Okay, so now let's take a look at this question. Quite long, this question, so we are going to try to utilize as much of the space as we possibly can and hope that we have enough room. So females on top, let's go the capital B with the X and capital V. See why it's really important to try to separate or show those small case and capital letters very differently. Capital B with the X and little v. So I'm going to put my little v with a hook. Little b with the capital there. So we're using the same process. And a little b with the X with the little v. Little v there. Okay, so that's the female across the top. Let's do the male now. So the male, big B with the X capital V. Big B with the Y. And again, in my opinion, hardest question with dihybrids that you're going to get there. Now, little b with the capital V, and little b with the Y. Okay, and probably beneficial to have lots of colors when we actually determine the phenotypes. Let's bring them together now. Long question, but let's go through it. Capital B, X with a capital V, X with a capital V. So that is going to be a red or wild type female. So you might have seen me say wild female, red female. Okay, capital B, X with a capital V, X with a little v. Doesn't matter as long as there's a big B and a big V on that X. It's going to be a wild red female again. Big B, little b, X with the capital V, X with the capital V. That's going to be a wild red female again. Capital B, little b, X with a big V, X with a little v. That's going to be a wild female, red female. Okay, my hand's getting sore here now. Capital B, X with a capital V and a Y. Again, if you have a capital B with a capital V on that X, I don't care what the other allele is, it's still going to be a wild red, but when it has a Y, that's going to give me a wild red male. Okay, and we do have to separate those guys when we do the phenotypes. Capital B, X with a little v and a y. So go back to my legends. As long as I have a capital B and an X with a little v, now we didn't show this in there, but if that's a Y, that is going to be a vermilion male. Okay, so as long as I have a big B with a little v, it's going to be vermilion. If I have two of the X's with little v's, it's a vermilion female. But in this case it had a Y, so vermilion male. Capital B, little b, X with a capital V and a Y. Again, if I have a capital B with a capital V, I don't care what the other allele is, that's going to be a wild red male. Capital B, little b, X with a little v and a Y, same thing. If I have a big B with a little v, that's going to be a vermilion, but with the Y it makes it vermilion male. Okay, bring them together again. Capital B, little b, X with a big V, X with a big V. As long as I have a big B and a big V on the X, it's going to be a wild red female. big B, little b, X with a big V, X with a little v. Always show the dominant allele first. Wild female. Of course, wild means red. Little b, X with a capital V, X with a capital V. Okay, so go in there. If I have two little b's, right? Two little b's with a capital V, doesn't matter what the other allele is, as long as there's a capital V, that's going to be a brown, and because this one has another X, female. Okay, little b, X with a capital V, and we already said it didn't matter what the other allele is. In this case it was a little v, but that's still, as long as there's a little v's and a capital V on that X, that's going to be another brown female. So, tough question, eh? Very time consuming. I'm sure you've called me a few names since we've been doing this question. So capital B, little b, X with a big V and a Y. As long as I have a big B with a capital big V on the X, that has to again be a wild. In this case it has a Y, so it's a wild red male. Okay, capital B, little b, X with a little v and a Y. So if I have a big B with a little v, that's going to be vermilion. And because it has a Y, that's a vermilion male. Little b, so little b with an X and a capital V and a Y. If I have little b's with a capital V, right, capital V, it's going to be brown. But because it has a Y, it's going to be a brown male. And a little b, X with a little v and a Y. This is that one we did and we added on over here. So if I have little v with a Y, that's going to be a white male. So big ratio going on here. Let's do the one that has the highest ratio first. So what I'm going to try to do here is get a color. Make them, let's say, blue. So let's look at our wild females. There appears to be lots of wild red females out there. So 1 wild female, 2, 3, 4. So we got to skip that line because those are all males. 5, 6, and those are brown. So I see that being the highest. So we're going to have 6 wild red females. Okay, and then we just systematically go through this and see if we can find the next highest ratio. I think there's quite a few wild males out there, so let's put him as a different color. Let's put him black. So again, when you're looking at the males, just look at those 2 rows. Wild male, that's 1. Wild male, that's 2. Those are vermilion. Go down to the next level. That's 3. Any other wilds? Nope. So that's it. So we have 3 wild red males, and you do got to separate males and females of the same phenotype. And let's go see if we can find any vermilion. Do we see any of I think that's the next highest here. Let's look at vermilion. Vermilion male, male, male. That looks like quite a bit. So let's change the color on this and make this one, let's say green. So we got a vermilion male here, vermilion male here, vermilion male here, and that's all I see. So that is going to be a verm male. Do I have any vermilion females? I don't think I do, right? Let's look at our females. Those are all accounted for. Nope, no more vermilion males. So let's go to the next one. Let's take a look at any browns. Okay, do we see any brown female? None on that line, they're all accounted for. Let's look at this line. I see a brown female here, so let's change colors. Put this purple. Okay, so brown female, none on there, so that's going to be 2 brown females. And then what do we got left? We got just 2 males, right? So let's just grab those. One of these are going to be a brown male. And one of these are going to be a white male. Okay, just ran out of room there. And I think everyone else is counting for. So we have a 6, 3, 3, 2, 1, 1 ratio. And those are the different phenotypes for each one of those. Long question, any issues, give me a call. Again, very complex, probably the most complex of the polygenetic dihybrids, and that probably not going to see a lot of these type of questions, but that's how you do it. If you have any questions, give me an email. Thanks, guys.